So let's listen to who this guy
my
is.
background, I've been in genomics for about
Talk to me
25
about
years.
sound.
I started at the Whitehead Institute in Cambridge, Mass. This is with Eric Lander working on the Human Genome Project. I was the team leader for R&D there. We built all the robotics that's in that Nature paper for the floor that does all the purification and sequencing for that project. And around 2000, I spun some of that stuff out. There was several patents we filed on magnetic bead isolation of viruses and DNA from E. coli, and that stuff got spun out to a company called Agincourt. Agincourt became one of the larger sequencing providers in the country at the time, and it eventually got acquired by Beckman Coulter for a lot of the viral purification tools we had. We were building these RNA and DNA purification kits using these magnetic beads, and so they needed that, and they needed to bolt that onto some of the automation they had. So they came and acquired the company and spun out one of our R&D projects that was building the solid sequencer at the time. That got acquired a year later by Applied Biosystems. They brought that sequencer to market. I spent about 5 years there working on that platform, competing with Illumina, and then also brewing up other ones. We ultimately acquired Ion Torrent, which makes these chips right here. It's another one of those little sequencers that runs on a chip. That I didn't invent any of that. I was just someone who, we saw the technology and pulled it in, and Jonathan Rothberg's team kind of carried a lot of that weight, but our team at Beverly also, about 100 of us jumped on making that platform sing. And then after that, I kind of just jumped out to start, we need to do something different, and I had a non-compete, and now the company was Life Tech, and it pretty much meant I couldn't work anywhere in the biotech space because they had fingers everywhere. So I started working in the cannabis space, doing sequencing cannabis genomes, trying to get more of these compounds public, if you will. What I mean by public is there's a lot of the things that you're seeing in COVID right now have been plaguing that industry for decades, where the science is funded primarily by government, and it crafts a certain narrative about harms. And so I was noticing that plant was severely underappreciated, and we just put its genome public. And since then, we've been building picks and shovels. We sell SNP chips and PCR kits to that industry to help them breed and help them track down viruses and track down different microbes that may affect the yield of the plant. So that's my background. I did do a little bit of clinical sequencing as well for 5 years. So I know some of the CLIA rules and regulations and how you should be doing PCR and not be doing PCR in clinical settings. And that got me dragged into the Corman-Drosten review. And then I've been doing some work very recently on just looking at the mRNA structures with Peter McCullough and a few others.
So that's Kevin's background. If it's not very clear, he worked on the Human Genome Project. He's been big into magnetic bead technology, which is in a wide variety of applications in all different biosciences, especially academic biology, because you can have beads that find their way to certain places and then and bring things with them. You can use magnetics to move them around. There's lots of different ways that those things are handy and were used for a very long time, in addition to the sequencing technology that they applied. This is kind of out of order here because he's now doing this. He was on the Drosten rebuttal, which was a PCR, a paper written by him and Michael Yeadon and about 8 other scientists that was objecting to the WHO protocol, which was written by a German named Drosten. And so we're looking at a very skilled, very wide background molecular biologist, and we're going to ask him a few questions about the RNA that they are injecting, but also we're going to start out with the PCR and talk a little bit about the PCR test and what's missing and what's not. We're not going to go into the details of how PCR works because both of us know how that works, and it wasn't good for the conversation. If we need to do that, we can do that on another day and revisit this. So what's the status of the rebuttal was the first question that we got to here in the discussion. Do you want to expand at all? Has anything happened with the Corman-Drosten thing at all? Or?
Well, so that thing we put that public. It got, the first one got attacked pretty heavily, and we put it public. So we wrote like another 60 pages trying to explain all the references behind what we have. And that thing barely ever got read. I don't even think Stephen Buston read it when he reviewed the paper. So there is, if you go to my Twitter page, go through the story that's on the top of that page. What's really interesting.
It doesn't actually work right now. Whoops. do you want to expand at all? Has anything happened with the Corman Jorston thing at all? Or
Well, so that thing we put that public. It got the
that
first one
media
got
doesn't
attacked pretty
load
heavily
what
when
he
we put
wants me
public,
to
so we
look
wrote
at. It
another
doesn't
60
load anymore. I
pages
think it
trying
got
to explain
removed.
all the references behind
Right.
what we have. And that thing barely ever got read. I don't even think Stephen Buston read it when he reviewed the paper. So there is, if you go to my Twitter page, go through the story that's on the top of that page. What's really interesting about what's pinned up there is that the people who are trying to attack that paper are on record in court cases before attacking Stephen Wakefield for the same damn PCR problems that are in the Drosten paper. And they got paid £500,000 to do it. Stephen Buston got paid by Glaxo to take down Wakefield. And he was attacking him for not having internal controls in the PCR, for not, which, Drosten had no internal controls. At least Wakefield tried to have an internal control. It just wasn't performing appropriately, but his assay had one. Drosten doesn't even have one. And then a lot of the other things that we've, the protocol wasn't very clear. A lot of the critiques that we put forward in terms of primer dimers and everything else, those things exist in the Drosten protocol. He was on record in a legal case attacking Wakefield for those same things. So that's the reviewer that Eurosurveillance could pull up. It's the only one that went public. They wouldn't put any of the reviews public. So we just have to go over the fact that he leaked some of this information on a podcast, that he was a reviewer and he didn't even look at the primers during the review, which is what our whole 60-page addendum was attacking. So I don't know. I don't have much hope that clarifying that's going to end the pandemic. Back then.
No, I agree. I agree. And so much of that is technical that for the vast majority of the people that could in theory be convinced, a simple hand wave from the right person and that's gone. But you brought up something twice in that description that I really wanted to ask you about, which is, are there PCR tests that have what you would call a appropriate control?
Yeah, there are other tests out there that have internal controls. They go off the RNase P gene, and that helps them tremendously. But what we're not seeing the industry do is pull out all the live dead PCR technology, which has existed for probably a decade. It's been refined throughout a decade. This is the capability to get PCR to tell you what's infectious virus, which is non-infectious virus.
I see.
And they're not doing that for a very explicit reason, because that would cut their revenues by an order of magnitude. I mean, if you look at just the how long people are infectious versus non-infectious, it's either 5 to 10-fold. And now they tell you don't get PCR tested for 90 days if you've been positive, right? Because you might still be positive from the last infection, but you're not.
How many academic molecular biologists in your imagination should actually, by their own experience, know that these tests are one inadequate, and two, that there's technology that already exists that would have been better anyway? How many people should really know that? Shouldn't there be thousands? Or
there should be. Yeah, I think anyone in the molecular biology space or genomic space knows that there's live dead tools out there. There's even stuff published on SARS with this, that you can do live dead, viability PCR on these things. So it's not like, if you just Google search it, you'd find it. But the problem is that there's so many people they sweep up in that tail of PCR positivity when you're no longer infectious. And then that explodes into a contact map of everyone else you're in contact with. So they're sweeping in an enormous number of clinically false positive leads. Yes, technically the RNA is there, but they're not infectious. And so I call that a clinical false positive. And that's probably the bigger problem than all the Drosten things are more like analytical false positives, right? Like your primers hit the wrong shit, or you haven't proven to us that this doesn't hit this doesn't hit other viruses like the flu. Those are things you can get in the weeds on with molecular biologists. But I think the real thing that's damaging society is that the way PCR is currently calibrated, which is very rarely against vero cells and very rarely through a viability check, even when it's deployed, you have this long tail of people that are positive with RNA that aren't infectious. They get swept in, they get contact traced, all their contacts get PCR'd. It just creates this huge explosion of PCR activity that effectively just shuts down society.
As I understand it, a clinical false positive is not an asymptomatic infection. It is just the presence of the RNA. And depending on the test you're using, which as he's just said a minute ago, the Jorston test, the sequences and the primers that are used in the WHO test were not even proven to not overlap significantly with other coronaviruses. And so even if they had a good test, they also need to have this control, which is a gene that they expect to be there, an RNA transcript they expect to be there in a baseline quantity in every human. And then from that baseline quantity, they can then either see where the virus fits relative to that baseline. But if you don't have that baseline, then you don't have anywhere to put the CT of the virus. And so that's what he's trying to explain here. But in your space for botanicals and whatever, you have to have a pretty specific, I mean, you must have run quite a lot of checks to be sure that what you're testing for is really there and that you're not producing a test.
Well, I mean, that's what drives me nuts is these are clinical tests going on humans. We have more controls testing candidates. I mean, it's like there's an internal control on every one of our tests. You have to have, in order for you to have a viral load, which is what everyone's talking about, you have to have two different CTs. You can't just have one. You need to have the host CT and you need to have the virus CT. So you have a ratio of virus to host. When you just skip the host, you have no clue what that number means. There's been papers published showing there's about a 10,000-fold variance, 1,000, 10,000. It's a paper called Dadu, in swabbing your nose. That's the variance in the number of cells you get.
Can you do that all in the same tube then, or do you have two separate samples for?
Yeah, you put them in. You put primers in there that have fluorophores that go off in different colors. So and there are some PCR kits that do this in the coronavirus space. So I don't mean to disparage all of them and call them all Drosten, but the ones in the human space they measure RNase P, which is a gene that's human, and they compare that CT value to the CT value you get from the virus. Now you have a numerator and a denominator, and you can call a viral load. And according to the Mikey guidelines that Buston Drosten himself authored, spell this out very clearly. You don't have a viral load without two numbers. You need both. But there is a lot of PCR out there in the coronavirus space where they're just ignoring the internal control and just saying if we get any signal out to 37, we call it positive. Right. And they will refuse to sequence anything. I think the CDC only sequences stuff below 28, and I see other labs doing it below 32. So they don't really have sequence validation on the stuff past 32. All that stuff could be noise. It could be influenza. It could be other coronavirus.
And so what he's not really mentioning, maybe he's forgotten, maybe he just doesn't want it to be as bad as it is, but they've gone out as far as 42 and 45. So if 32 to 35 is noise, then 35 to 42 is definitely noise. So we're really at a pretty severe place in history if the first year, and that's why I showed that timeline, was because that whole timeline before Hert first speaks up about the enrichment for escape, there's a whole year where we're using PCR as willy-nilly as possible, classifying everyone with a PCR positive within 28 days of death has a COVID death. And all of those numbers are still part of the 800,000 people they claim to have died over the last 2 years for COVID. It's very important to remember that for almost the first year of the pandemic, anyone who tested positive by the standards you just heard described as being horrible, anyone who tested positive within 28 days of their death would have been considered a COVID death and is part of that total, which is being rammed down your throat every night by Fauci, Walensky, and the rest of the crew. And so what's the difference with this RNA from the viral RNA? What's the difference from this RNA from a regular RNA? What do these things cause? What can these things do? From a general biologist perspective, has been on my mind for a long time, is the idea that Moderna and Pfizer have told us that they have this sequence and they can make many good copies of it and then put it in our cells, and they'll make very many good copies of a protein that will harmlessly provoke an immune response that we can rely on in the future. And a lot of that seems odd, but the most odd of it was this calling it transfection or vaccination instead of transfection. And I always understood transfection as being something that, I mean, when we did it to our mice, it was an endpoint. After 5 or 6 weeks of having transfected a particular, let's say, optogenetic protein in the brain, that area of the brain is also going to be a little bit
unhappy.
Yeah, unhappy. Let's say it like that. Yes.
Yeah, if you overdo it with the lipid nanoparticles, they become toxic to the cells. So you can't. Usually, you have to go in at low MOI, low multiplicity infection, in order for you to get those transfection systems to behave. I've done some mammalian transfection, and I can't say this about the lipid nanoparticles they're using because they've modified them, but the ones that we were typically using from Invitrogen, the surest way to destroy your cell lines was to put too much of it in there. So I'm very skeptical of what they've claimed. They've claimed they have these mRNAs that get transfected, and then that they make this perfect spike protein. They've never shown that. I mean, unless your audience can find, I really hope someone can prove me wrong here, because there's now like so many millions of papers of SARS-CoV-2 out there. It's very hard to dig through the literature and find exactly.
Oh, I've been looking.
Yeah, but I cannot find.
I've been looking for if anyone has found like dose, if they have some range of what they expect to go into circulation or what they expect to be produced. And the other thing that you've written a paper about recently is the obvious unknown of post-translational modification, and then how the protein itself is folded, given the fact that it's not the actual sequence. They've altered the sequence in such a way.
Yeah, so that is I think that's criminal in that it's technically a prodrug, right? Because you're giving a sequence that you're transfecting into cells, and you're asking your cells to make the final drug without ever having to declare what the final drug is or prove the fidelity of making that final drug. They got away with a hall pass on that. Like, if this
Did you hear that? Let me just say that again. Say that again.
Right? Because you're giving a sequence that you're transfecting into cells, and you're asking your cells to make the final drug without ever having to declare what the final drug is or prove the fidelity of making that final drug. They got away with a hall pass on that. Like, if this were some other drug that was a prodrug that your liver modified and turned into another drug, you'd have to have all these P450 studies. You'd have to have demonstration.
That's a really wonderful way to explain it. I think a lot of people will, even some nurses and stuff would understand that if you point out that if this was a prodrug that had to go through the liver first, then you would have so many more. Let's just listen to that home run statement one more time.
That is, I think that's criminal in that it's technically a prodrug, right? Because you're giving a sequence that you're transfecting into cells, and you're asking your cells to make the final drug without ever having to declare what the final drug is or prove the fidelity of making that final drug. They got away with a hall pass on that. Like, if this were some other drug that was a prodrug that your liver modified and turned into another drug, you'd have to have all these P450 studies. You'd have to have demonstration.
That's a really wonderful way to explain it. I think a lot of people will, even some nurses and stuff would understand that if you point out that if this was a prodrug that had to go through the liver first, then you would have so many more checks and balances and tests and trials that would have needed to be done on all stages of that before.
You have to prove what the final drug is.
Right.
The final drug. They have no evidence that they're making a spike protein off of this mRNA. I mean, they have a couple. I shouldn't say no evidence. There's papers out there where they take these things and they express them in other hosts.
Right.
And I've seen a couple 3D, crystal structures of them. They've all used modifications of it. Like, they put a twin strep on one end so they can purify these and crystallize them. Or they're not using the pseudouridine template. Sometimes they're putting them in DNA vectors and getting them to express. So the actual mRNA getting injected into people, and what actual peptide sequences that those mRNAs manufacture in cells, I can't find literature on. I can find some people sent me material that were from Pfizer's disclosures, I think, to some European government agencies, which I can forward to you if you want to link into this chat. But they basically show that they have all types of problems in multiple different bands. When they go and try to express these in vitro, they get all types of fragmentation that occurs, and they don't know if that's fragmentation of the virus or stall points in translation. And the banding patterns they see on gels are smears. And there's questions on them having to clarify, why are those smears not the right size, the anticipated size? And that's all we can see from some of the regulatory disclosures. So what has me worried about what they did is they took a sequence that was already codon optimized by a virus adapted to its host and decided to make it different. And we know this is now the most toxic protein probably in the virus, right? So if a virus has evolved a certain codon structure for a toxic peptide, it probably has got the codon structure right, and that it needs to make sure it's not lethal to the host so it can still survive transmission. But they went with the route of supercharging this by codon optimizing it so it would express even more in human. I mean, this is what I think I do. Whenever you want to hyperexpress a protein, this is just what we do. We codon optimize it. We put it behind a really heavy promoter, toss it into some cells that are going to be sacrificial, and just jack the expression of it up. But you're doing this in cells you know you're going to kill, and then purify as much of the damn protein as you can. That's not necessarily what you need to do to dose humans. You shouldn't be necessarily aiming for maximum expression, because when an RNA gets into a cell and starts making maximum expression, all types of triggers go off on the interferon system, being like, this could be cancerous. Like, what the hell's going on here? You could get apoptosis because you're expressing way too much protein in a particular cell. So they did a lot of things to jack up the expression. They put a beta globin promoter on there, which is a pretty aggressive promoter. They then codon optimized it, which didn't need to be done, in my opinion.
Okay, I'm going to play this again because there's a lot of questions here, and it's good to listen to him again because he's an excellent speaker, but some of the audio is not perfect because this is just from Zoom. But the first thing to keep in mind is that codon optimization is making synonymous changes in the 3 nucleotide signals for the amino acids to optimize them for maximum translation. And that has to do with making the codon the right codon, and that's a very complicated thing to explain, but usually there are more than one 3-letter combination for each amino acid, and there's one of those 3-letter combinations that is the fastest one for a ribosome to read. And so the first thing they did was they went through the spike protein of the virus and they took each amino acid codon, and rather than using the codon that the virus uses, they optimized it for maximum protein production. Now, that should not change the protein that comes out. It should just change the amount of protein that's made, in theory. And then after that, they've done something else. So let's just listen to that prodrug thing again. What's your way?
Yeah, so that is, I think that's criminal in that it's technically a prodrug, right? Because you're giving a sequence that you're transfecting into cells, and you're asking your cells to make the final drug without ever having to declare what the final drug is or prove the fidelity of making that final drug.
And so prove what the final drug is one thing. Like, let's say, for example, the prodrug is the pencil, and then the eraser gets taken off, and now this is the active drug. This would be the drug. And then you have to prove mechanistically that the liver removes this part of the molecule, and then this is the active molecule. And so they have not proven that whatever they put in you produces a spike protein. And in fact, what he says about the gels is that it's a smear, because what would normally happen when you make a gel is that you have a mix of proteins in the thing that you want to separate, and then you put them in the gel, and the big proteins go slow and the small proteins go fast, and then when you stop the gel, you get a bunch of proteins in one band up here that are big proteins and don't move very fast through the gel, and then you get another band lower than that, because these proteins are all tiny and they are all the same weight too, and so they show up at a different place on the gel. And so what he's saying is that when they did this, what they see is a smear, meaning that it's not like there's an S1 or an S2 subunit and an S1 subunit. And you can see very clearly that there are 2 proteins that are produced, but it's a smear because there are so many errors made. There is no clear banding. And so all kinds of random fragments are produced when these RNAs are translated. And then finally he says something else. Maybe I'll pause it in the middle and explain that. I can't remember.
They got away with a hall pass on that. Like, if this were some other drug that was a prodrug that your liver modified and turned into another drug, you'd have to have all these P450 studies. You'd have to have demonstrations.
That's a really wonderful way to explain it. I think a lot of people will, even some nurses and stuff would understand that if you point out that if this was a prodrug that had to go through the liver first, then you would have so many more checks and balances and tests and trials that would have needed to be done on all stages of that before.
You have to prove what the final drug is.
Right.
The final drug. They have no evidence that they're making a spike protein off of this mRNA. I mean, they have a couple. I shouldn't say no evidence. There's papers out there where they take these things and they express them in other hosts,
Right.
And I've seen a couple 3D, crystal structures of them. But they've all used modifications of it. Like they put a twin strap on one end so they can purify these and crystallize them. Or they're not using the pseudo-uridine template. Sometimes they're putting.
Okay, so sorry about that. So I want to make sure you understand that what he said there is when they proved that the RNA could produce spike protein, they did it with RNA and lipid nanoparticles in cells, and they did it not with the pseudouridine transcript, but with a normal RNA transcript. And that's going to come up in a minute, but you really need to hear that because that's extremely important. Then you would have so many more checks.
They're putting them in DNA vectors and getting them to express, so the actual mRNA getting injected into people and what actual peptide sequences that those mRNAs manufacture in cells, I can't find literature on. I can find some people sent me material that were from Pfizer's disclosures, I think, to some European government agencies, which I can forward to you if you want to link into this chat. But
I'll try and
they
get
basically
them.
show that they have all types of problems in multiple different bands. When they go and try to express these in vitro, they get all types of fragmentation that occurs, and they don't know if that's fragmentation of the virus or stall points in translation. And the banding patterns they see on gels are smears. And there's questions on them having to clarify why are those smears not the right size, the anticipated size. And that's all we can see from some of the regulatory disclosures. So what has me worried about what they did is they took a sequence that was already codon optimized by a virus adapted to its host and decided to make it different. And we know this is now the most toxic protein probably in the virus, right? So if a virus has evolved a certain codon structure for a toxic peptide, it probably has got the codon structure right. And that it needs to make sure it's not lethal to the host so it can still survive transmission. But they went with the route of supercharging this by codon optimizing it so it would express even more in human. I mean, this is what I think I do. Whenever you want to hyperexpress a protein, this is just what we do. We codon optimize it, we put it behind a really heavy promoter, toss it into some cells that are going to be sacrificial, and just jack the expression of it up. But you're doing this in cells you're going to kill. and
Chancellor Sopat is correct. A smear suggests it's happening continuously along the RNA molecule, and we're going to talk about in the next clip. He's going to explain how that happens. So you're right on with that. Just let me finish this clip. Will be the next one.
And then purify as much of the damn protein as you can. That's not necessarily what you need to do to dose humans. You shouldn't be necessarily aiming for maximum expression, because when an RNA gets into a cell and starts making maximum expression, all types of triggers go off on the interferon system, being like, this could be cancerous. Like, what the hell's going on here? You could get apoptosis because you're expressing way too much protein in a particular cell. So they did a lot of things to jack up the expression. They put a beta globin promoter on there, which is a pretty aggressive promoter. They then codon optimized it, which didn't need to be done, in my opinion. And that creates all types of other problems. When you codon optimize it, you don't know that the translation is going to go at the same speed anymore. It may slow down due to all types of knots in the RNA you created by doing this. Like there are quadruplex G structures now that we know in the RNA that are much more frequent in the messenger RNAs than they are in the actual natural virus.
Does that make error rate go up then too, or?
It makes translation error go up. It also attracts other types of regulatory proteins to the molecules, because quadruplex G structures look like telomeres, and so telomeres are involved in sensinins. If you look at your telomeres, they're all quadruplex Gs, and so those things recruit proteins that are involved in the life cycle of the cell, that are involved in cancer, and that are involved in sensinins. So there's actually proteins in SARS that are attracted to. There's the NSP13 protein is attracted to quadruplex Gs, right? So they now have much. I think there's like 4 in the natural virus. It goes to like maybe 9 and into the teens on the Moderna and Pfizer vaccine. So they're creating a lot more of these stall points in translation and other biological motifs that might alter the biochemistry.
The way that he explained it in one of the clips that I don't show is it's kind of like a telephone cord from the old days when it gets secondarily knotted up and you can't pull it out anymore. A lot of these G quadruplex structures result in a complex three-dimensional structure in the RNA molecule itself, and it can make it very difficult for it to pass through the ribosome, and it can cause skipping in translation, frame shift, etc. So that's interesting. Now, here's where it gets really cool. Here's where I was absolutely blown away. This is about the pseudouridine. This is about the replacement of uracil with pseudouridine and what it can cause and what we know about it from previous work. It's just amazing.
So the other thing that they did is they put a universal base in there known as pseudouridine. All right. Many have probably heard about this. The patents go to UPenn, and
I've never heard it called a universal base before, though.
It's a degenerate base. Yeah, so it binds to, they won't call it that, but I've built DNA sequencers on degenerate bases, and those things were on our radar for a reason. Yeah, so it binds to, it has pseudouridine being uracil, should have, it's like a thymidine replacement, okay? So it should have one of these AT pairs.
Right.
And unfortunately, this thing also binds to G. It also binds to itself. It also has some binding to cytosine. So this means that it's a very, very sticky base. It can flip around and bind. If you actually look at its glycosic bond on the
But
base.
I don't want to sound like an idiot, but this is the first time I've ever heard this. For me, I always just assumed it was a chemically different substitute for a single base.
It does. It replaces it. They're replacing all of the U's with this because it's more RNase resistant. Okay, so it doesn't degrade as much. So so you've now got an RNA. You've codon optimized it. You put a heavy promoter on it, and now you also took away the brakes. The brakes are usually RNases that come in and chew up these messenger RNAs. But they put a long poly A tail on this, and they put these pseudouridines in there so the RNases can't touch it. Now, there's a consequence to doing that is when you camouflage these things from RNases, you also start triggering a lot of the toll receptors to get malfunction, like toll receptor 3, 7, and 8 get all screwy with pseudouridine. But that's a separate topic we can bench for later. The thing I want to get across is that
sorry. I'm not trying to mod the chat at all, but just try not to use racial terms that make me have to mod the chat. I just don't want it. I'm sorry. It's not that I don't understand. I'm a multiracial American, but I just
base that they have in there is not very specific. And so when you don't have specific binding, you can get all types of misbinding. You can get all types of hairpin formations to form. But more importantly, this will create errors in replication in DNA, but the errors that are actually orders of magnitude more frequent are errors in translation. Your tRNAs then have to come in and read this stuff.
RNA to protein.
And when you have universal bases in there, they get all confused and the translation errors go through the roof. And there's a couple papers I can point you to.
Bumping your microphone or something.
Oh, I am. My hands are on it. Sorry, there must be.
It's okay.
Sorry. So when you have this universal base in there, it not only confuses polymerases, but it more readily confuses the tRNAs and the ribosomes. They have a much harder job of having to put these triplet codons in there. And when one of the bases is mismatching to multiple different bases, the translation errors go through the roof.
Right.
Now, there's a paper on there that tries to measure these translation errors with pseudouridine, and they measure they can detect it when there's one pseudouridine in there. And from what we know from these mRNAs, every uracil is replaced with these. So they're getting codons invariably that have multiple pseudouridines underneath these tRNAs. There's no way that thing creates high fidelity.
You just said that they could detect the translation errors when there was only one?
Yes, and these things have replaced all of them. So
that's a huge statement. Again,
yeah.
I'm learning a lot of this for the first time. I did not realize that this was this awful. So coming back to Chancellor's comment then, right? Sopat's comment about how the smear is generated. The smear is generated because every uracil is replaced by a pseudouridine, and every one of those places is a potential translation error, because in papers where they look at the effect of pseudouridine in translation, they can see the effect of a single replacement. And again, that single replacement would result in a fragmentation of the protein such that rather than translating only pencils, right? This would be the translation product of a good RNA. And then if you put a single pseudo-uridine here, then the translation could stop or be interrupted there, such that you would get some products that look like this and some products that would be these products, or just these, or just these. Because at the pseudouracil, you can get interruption of translation and errors or frame shift, etc. And all of those things then will result in a different length protein as a result of the output. The more errors you make, the more places you can make errors, the more different length proteins you can get, and therefore you'll get this smear on the gel, which we described earlier. Wow. Oh, is that it? That was the that's funny. Okay, so the next one, and then we're going to get next to this one because, again, you heard it implied earlier, the stop codons, right? The stop codons. I'm sorry, what is this dude doing? Does that work? I think that works. I don't know if that worked. Anyway, whatever. I don't want to be too distracted by it, but he's a dork. Anyway, here we go. Let's do this. This is the stop codon part that he just mentioned. It's really nice. Really nice. And it's a great explanation by him. I just think that he's a really nice guy and he really knows what he's talking about much more than I did.
Here's another thing that's really spooky about at least the Pfizer one, is there's some papers out there that show when you put pseudouridine in the stop codons, the ribosomes read through it. And they sometimes frame shift on the way through it. It means they keep translating the RNA sequence that's past the double stop codon. So you have to ask what's on the 3' end of the Pfizer UTR, like what do they have back there? And we went and scanned all those things. And there's actually a human peptide on the other end of that could get expressed as a chimeric protein. We've got no evidence for this in vivo. This is just all in silico stuff that we're looking at. Like if pseudouridine ablates that stop codon, what does it read into? And it reads into a fairly long peptide that has some homology to GP130, but we don't know much more about it. The data in NCBI is pretty sparse on whatever that thing is, but some of that's in the paper we put together with Peter McCullough as to what the human peptide is.
Are you following this? There is a human peptide or a human-like peptide coded after the final double stop codon, and since they're pseudouracils, chances are our ribosome can read right through that and translate that protein, and nobody's talking about whether that protein is produced or what that protein is, but it has homology to GP120, which is a...
what does that mean? If there's some error rate here, and I'm not suggesting every single spike protein that comes off this mRNA is going to do this. These error rates happen at frequencies that are very low, but you have to keep them in context to how many molecules they're injecting into you. They're injecting trillions of these things into you, right? So those numbers times an error rate that some I've seen 10 to the -4th, 10 to the -5th rate, they matter. They matter. You could get chimeric proteins that come out of this that have human SARS chimeras. And what does that do? Does it create autoantibody issues? Does that create autoimmune issues? The long story short is these are all hypotheses that haven't been explored. They're in silico, but you do have to ask, why did Pfizer and Moderna and these people get away with getting the prodrug certified without ever having to show their hand on what the hell these things make in human cells? I've dug up a couple papers in that paper with Peter on there's a couple other images in papers that look like this could be happening. Like there's one great paper out there, I think from Bansal, that looks at like spike proteins that are expressed in exosomes, right? In the blood. They showed them like 4 months later. Really interesting paper. They do have some like SDS page images of what of those exosomes, and they're smeary. And then there's another paper we found from Jiang et al. that showed these showing fragments, and the fragments that were getting expressed of the spike protein, they weren't all full length. They were different based on the cell fraction they isolated them from, right? And I think that was even with the DNA vaccine that didn't have the pseudouridine problems, right? So you can imagine that would get even worse if you had all these other complications put in there. So my concern is we don't really know what these vaccines are producing. We don't know the fidelity of it. We suspect there's some spike protein because there are motifs that look like spike proteins because antibody studies, but we only have antibodies. So it's kind of like the people, blindfolded doctors touching the elephant, right? You got some similarity, fine. But we don't precisely know the diversity of what's getting expressed in humans. And we should be concerned about that because these LNPs don't target particular cell lines, they go everywhere. So that's a much broader problem. If you knew that, okay, we're expressing some fragmented spikes, but we've got them well characterized, the data's public, and they're going only to cell lines that the virus sees, like ACE2 expressing cell lines or CD147 expressing cell lines, fine. You've got an understood complexity of the problem. When you start using these LNPs, they transfect every cell they touch. So now you've got to worry about not just spike, what does spike toxicity do in those? What does all of the degenerate products of low fidelity spike expression do to many more cell lines?
So understand how he's adding to the complexity of the problem when he explains to you that the LNP, the lipid nanoparticle, if it targeted ACE2 receptor expressing cell types, at least then the target of the LNP would be known and it would be relevant to the virus and relevant to vulnerabilities as prescribed by how we understand the virus. But instead, it goes everywhere and it can express in any cell type, many of whom may not have vulnerabilities to the virus. And so then what happens if a cell that wouldn't normally express the spike during a infection starts expressing trillions or billions or millions of copies of the spike and spike fragments because it's very low fidelity expression of whatever transcript gets in there. Low fidelity means high variability. It means a lot of garbage, a lot of wasted time. And those RNAs aren't going to get degraded because of the way they've been chemically altered. It's a spectacular, damn it. You just hit the wrong keys.
But we don't precisely know the diversity of what's getting expressed in humans. And we should be concerned about that because these LNPs don't target particular cell lines, they go everywhere. So that's a much broader problem. If you knew that, okay, we're expressing some fragmented spikes, but we've got them well characterized, the data's public, and they're going only to cell lines that the virus sees, like ACE2 expressing cell lines or CD147 expressing cell lines, fine. You've got an understood complexity of the problem. When you start using these LNPs, they transfect every cell they touch. So now you've got to worry about not just what does spike toxicity do in those? What does all of the degenerate products of low fidelity spike expression do to many more cell lines? It can really blow up like the complexity of the problem and even perhaps the symptomatology, right?
This is blowing my mind because it didn't really the extent to which this adds variables is really breathtaking, actually.
It's been frustrating me because I've heard so many arguments online that, it's just the spike protein that's identical to what's in the virus. No biggie, right? So your symptoms, your adverse event profile should be a subset of what you get from the virus because we're giving you a subset of the virus. We're not giving you the full replicative competent virus. I don't think that theory should be accepted blindly because we're injecting these things into more varied cell lines. We are injecting them, I think, at a higher concentration. I mean, the way, how do you read those antibody studies, right? You see that they have higher antibody expression against spike, right? I don't read that as like a better immune response. I read that as you're expressing more spike. And so the antibodies are probably a proxy to the expression level of spike. And is that a good thing? Do you really need massive amounts of spike protein to build an immune response? Because we have to be careful here. Spike's the toxic peptide, so we're in this window like cancer treatment where we're trying to kill the host cells
And am I
cancer.
right? And am I right in my understanding, though, that actually, if you go with this idea that antibodies are neutralizing the spike and that's how you build immunity in their cartoon world, then aren't they only really concerned about antibodies that have some specificity for the receptor binding domain? This leaves an awful lot of useless epitopes in that.
It does, yeah. And I don't know if you saw that recent paper that when they serially applied the same vaccination, that the actual antibodies got more and more specific to the
Right.
Wuhan type 1. So they're kind of making matters worse by boosting.
So we're going to get into that in a minute, but we're just ending it on that one note there, right? That he's also seen the papers that show, from the perspective of the spike protein, it looks great. We're making more and more memory to the spike, but it's actually redundant memory to epitopes that don't neutralize the virus. And so it's a red herring. It's an illusion in terms of what is actually happening when you are boosting somebody. You are buying more copies of the same book, and the book has already changed. So it's really not good. In this final one, we're going to talk a little bit more about what happens, and we're going to just listen to the talk as it rolls out, and then I'll summarize and I'll say goodnight. Oops. That was pretty fast.
Patients are less tolerant to future variants by boosting.
Right.
So
Yeah.
there's a host of problems with what they're doing on that front. But I hope someone can prove me wrong and find more papers, anything related to what proteins actually get expressed. I would have thought that if we have 5 million SARS genomes in NCBI, and maybe there's 2 million NCBI, but there's 5 million in GSET, right? How many sequence QCs do we have the lots on these vaccines? I can find one, like from Andy Fire's lab. And they didn't even put out the raw reads, they put out the consensus sequence. So you can't really dig into some of this nuance on how many variations are actually in there. So you would think there would be sequence QC lot to lot in all these vaccines. You would also think there'd be evidence of when you transfect this into humans, what's the peptide that comes out with mass spec profiles in the peptides, like protein sequencing. Why don't we know precisely what these things are? The protein sequencing gives you some information about what's going on. It doesn't necessarily tell you how things fold. I mean, one of the challenges in codon optimization is you do change the rate of translation when you do this. And there's lots of papers out there showing that when you change the rate of translation, you can alter the folding. It's particularly true when you hit double prolines, and there are a lot of double prolines in the spike protein, right? So those things end up stalling translation, and your codon usage really matters across those things. So,
but it's don't you find it also so frustrating that there are so many people that seem to have as much sort of intimate sequence gene type data, not data, but knowledge and understanding as yourself, but then focus it all on proving that the virus didn't come from a laboratory, and look at the sequence of the virus and wax intellectual about all of this stuff, but not once turn this same keen eye to the PCR tests, to the vaccine, or to the transfection, and think the same with the same sharp edge. I just don't get it.
I think it's a hierarchy of funding that drives all that. That you'll see, whether it's funding from NIH, NIAID, or from Gates, that all of those actors never want to attack the narrative. And if you start raising the questions like, hey, we've injected 1 billion people with this, shouldn't we have more sequence data on the actual vaccine? Like, you guys are doing a great job sequencing all those viruses and putting them public, but when are you going to turn your attention to what you're injecting into people forcefully? They turn that as into a conspiracy theory. They can't question it. They can't look at it. But if you were neutral and not in this and not funded, which I'm assuming both you and I are in that camp, and a variety of other genomicists are in that camp, we look at this thing, no, you should have an equal number of sequences on both sides of this fence. The virus is really important to see where it's going. But if we don't have, particularly when there's liability-free mandated injections going on and there's no QC, and if you ask for it, they mock you as an anti-vaxxer, that there's something really wrong there.
Yeah, I think that's a really nice way to put it too. I think that's are you in regular contact with Michael Yeadon since this, or not really at all, or?
We were in a little bit more contact when we were doing the PCR stuff, but I haven't. I've seen him on a couple. I'm on so many Twitter threads and whatnot. His name comes by. He got kicked off of Twitter, so I haven't seen as much of him since he's off of Twitter. But there's a couple other email threads that I'm on that he's there as well. But he did. He brought up some concern, I think, over what was it, syncytia 1 or something. It was a short amino acid sequence that he got a little bit flamed for.
Yeah.
But
he's still just out there every day trying to push it. I just didn't know. I think him and Peter McCullough and yourself and everybody who brings some kind of real life experience to this and isn't afraid to speak out about the incongruencies, I think is part of the solution. And I don't know. I've been following you for a long time on Twitter now since I figured out that you were one of the guys on that original Jorston thing. And I only did PCR I did single cell PCR and tried to prove that there were different nicotinic receptors in different neurons that we were pulling out of the brain. And so most of my methodology problems were just trying to convince people that we could get enough material to actually get a read. They were fine with the fact that there were nicotinic receptors there. That wasn't proving some kind of ghost or anything. But
you know from doing that how sensitive it is. You can
and how easy it is for somebody to say you don't have enough controls to know that you're saying what you're saying. And so when this started, it was pretty mind-boggling to me that rather than starting with a small needle that I had just pulled out of a brain slice and I had just recorded from, so I knew what kind of cell I was pulling out, you could stick a swab up in my nose and make the same measurement. That just seemed kind of impressive to me. I don't know.
Yeah, I've not seen the LODs on this that get down to single DNA or single copy levels, but they get close. They get down to 100 copies usually off those, sometimes 50 off of those nasal swabs. But the variance is with sodium wide.
Yeah, I see.
That you get the CTs that were in the Dadu paper were like 10 to 13 CTs from swab to swab on the internal control. And then you have everyone arguing over what the CT should be for the virus to cut. I'm like, well, if you don't normalize for how much you swab, you're never going to get that.
Mm-hmm.
Hold on, give me a second. I think my dog needs to go outside. Hold on, give me
I think we come right back after the dog here. So we're just going to close it up really fast. We're talking about a few other things here, and then it's the end of the video. But it just...
there's a host of problems with it that are just getting swept under the rug, and every time you raise this with people, they're just like, oh, you're whatever, anti-vax.
Can you explain if it's too complicated, you can't, please. But can you explain a little bit why, if the RNA folds in a particular shape that you're explaining, and then shape attracts proteins that cause senescence?
Well, they attract proteins associated with it. I don't know if it actually causes that yet. It's just there's keys in the literature that show when you form lots of quadruplex G formations in RNA, you radically alter the translation rate and you alter a lot of the other cell signaling that goes on. So quadruplex Gs are a different form of base pairing. Most base pairing is called Watson-Crick base pairing, where you get that nice double helix that you get about 10 bases per turn. When you get really GC-rich areas, what can happen is it's almost like imagine you're like those old phone coils you have that are perfectly helical, except it's once in a while they get inverted and they flip the other direction.
Yes.
That's kind of like a quadruplex G in DNA. They have these capacities to form Hoogsteen base pairing when they're GC rich, and then the structure of them changes into what looks like a knot. And those knots are stall points for translation, and they also attract a lot of other cell circuitry because they're signals that are regulatory in nature. You see a lot of these quadruplex Gs in promoters. You see a lot of them inside of the telomeres. So they're signatures to the cell that are involved in interferon response. So when you start making, when you codon optimize the virus's RNA into the human version of this, where the most of the A's and T's in the third base of the codon get swapped out for G's or C's. When you do that, your GC content goes up substantially. And when your GC content shifts that much, you get quadruplex G formation all over the RNAs. That's something that's not as native to the virus. The virus, actually, if you look at the quadruplex G structures in SARS, they're highly conserved. Like there's a paper out there showing they're conserved across 16,000 of the genomes they looked at. so that the virus really only has these in key places for purposeful regulatory reasons, and then they make a version of spike protein that then litters them with a shitload more of them. And I think I've got to dig up the exact paper, but they tripled in one of the vaccines and went up almost order of magnitude in another one. I mean, it went up to like, I think 16 or 19 in one of the others. So they've littered the RNA with these quadruplex Gs. They probably didn't notice this. I would argue they just did. If it was warp speed, they just put it through a codon optimization tool, because that's what you do to maximize expression, and they moved on. And so they're probably not aware of that. Oh, we put in all these secondary structures into the RNA. That's going to change the RNA's recognition by a lot of the toll receptors, and by a lot of the viral defense mechanisms that they have. A lot of these viral defense mechanisms actually key off of the secondary structure of the RNA to cut it. And that's changed now with the pseudouridine implementation and the codon optimization. So I think that's why we're seeing a lot of these papers emerge that suggest these mRNAs like reprogram the innate immune system, because these things go in the toll receptors.
The GC content, there's 4 bases in it, right? There's adenine, cytosine, guanine, and uracil. And in DNA, it would be thiamine, I think. So A and T pair together, and G and C pair together, or something. And so in RNA, the U replaces the T. That's it. But if in a particular codon, remember that each amino acid is 3 nucleotides, 3 bases encodes each amino acid in the protein. Those 3 base codes, often the last base is a wobble base. It doesn't really matter what's there. So you can substitute a G or a C there, and that'll make that codon read faster. And so they've done that with every codon. And over the course of thousands of amino acids, then you greatly increase the number of G's and C's in that code and cause the DNA to fold differently. That's what he's trying to explain. And he's saying that because they went through this specifically just to get more spike protein, then they didn't really think very carefully about what this would cause, but they had plenty of evidence that it was causing it. It's just maybe they didn't know or they didn't care. It's warp speed. I think he's being a little kind to them, but that's what he's explaining.
Are confused by them, and we don't know how long they remain confused, but there seems to be at least a week period of either lymphocytopenia or neutropenia that's occurring, and there's also some alterations in the ratios of CD4 positive and CD8 positive cells. Right? Those
and toll receptors are in all cells, right?
Toll receptors, they are immune. I don't want to say they're in all cells because I don't know actually, but they're in the gut a lot, and they're in a lot of cells that are I think on the epithelium layer to look for pathogens basically.
Okay.
Yeah, I don't know how they exist in the brain, but I know that they're heavily involved in leaky gut syndrome and monitoring what types of pathogens and the microbiome that you've got in your gut.
But how do they, if they're toll receptors, how do they interact with RNA that's inside?
So I think it's toll receptor 3 that looks for single-stranded RNA.
I see. Okay.
Yeah, and if that RNA has pseudouridine in it, I think it doesn't see it. The double-stranded RNA is another issue, which something Andy Fire's group did is they tried to measure the single-stranded versus double-stranded ratio of the RNA in the vaccines that they did sequence. And they found a very, very low frequency of the double-stranded RNA because the double-stranded RNA tends to trigger more of this innate immune response.
But the real reason that they told us on PBS NewsHour that they did all this pseudouridine was to avoid the
degradation.
The degradation. Only the degradation? I thought it was also the activation of the immune response in the...
Yes, it's both. And the question people are starting to raise is, okay, you evaded the immune response, but did you turn it off for a period of time?
I see. Okay.
Like, can you see other viruses now? We certainly see reactivation of other viruses post-vaccination.
Right. I see what you mean.
And of course, this gets even more complicated when you start considering breakthrough infections, because now you've got both to consider. You got to consider the biology of this modified RNA and how's it going to interact with the virus, which has other genes like NSP13. We know that one, it interacts with these quadruplex Gs.
But how are we going to differentiate between the I guess that's what that's what clinical trials should have done, but how can we differentiate in your mind between the effect of the never before tested complete pseudo uridine of all the U's in an RNA versus the effect of the toxicity of the protein itself? Because assuming that's also there.
Yeah, I don't know the answer to that. I think they need to do more in vitro translation and peptide sequencing to prove, like, what is this thing actually making? And you may not have to do that in human. You could do that in human cell lines first. That'd be a good step forward to see, okay, what does this produce in human cell lines? Ideally, you do it. You could capture the stuff in patients as well. If they're catching exosomes circulating in the human bloodstream with that Bansal paper,
right,
we should be able to capture those and sequence them. And see what the amino acid sequences of them are, at least have high fidelity. I don't know.
Sorry.
Enough about crystallization, whether you could capture enough through that method to actually get structure information out of it. That's probably a question for someone who's more in the structure space than I am. But that's
what I definitely sequence the protein. You could definitely sequence the protein if you could purify it.
I think you could, from the quantities. If you can see a band on a gel, they should be able to get that out and sequence. So the question is, will the mass spec systems that fragment these things be able to see, heteroplasmic translation, right? This translation where there's a small percentage of the protein mistranslated, or is that going to be harder for them to pick up? That's a question for a mass spec expert. I haven't done a lot of mass spec protein sequencing, but
what you see in the three-dimensional structure, would it lead you to believe that there are going to be like signature error points, or could you predict where?
I think so. I think you would probably see some of the consistent regions where they're fragmented or where they translation stalls, and you'll get spike fragments. Now, maybe some of those don't float around the exosomes because of that. I don't know. But you may be able to pick up partially folded spikes that would give you some understanding of, okay, we don't have one drug getting produced off of this prodrug. We have a library getting produced off this prodrug, and we have to consider that in the adverse response data that we're looking at.
And so what's your plan now? You're not going to get into this at all? You're just going to stick with your botanicals?
Yeah, I mean, I do this kind of as a hobby on the side because I'm as frustrated with everything going on in COVID as I can, but I don't have a lab that can work on this problem. Our company is all focused on making really good genomic tools for other industries, but I'm not vested in the COVID industry. So people have always told me, speak out if you can, because you're not. I don't get PCR revenue if COVID goes up or down. I don't have any. I don't have any vested interest in vaccines or any of this, so I can view this from a more neutral perspective. But I'm doing this mostly because my kids are affected by all this. It's been 2 years; their school's been totally blown up. My eldest son never graduated from high school. I mean, he technically graduated, but we never saw him. He never had that experience. All of these things in their life have just been turned upside down. They're having to wear masks into campus at universities. The same week they'll have a 60,000 person football stadium filled with people without masks, and they all go out and then go to their classes in masks. It's making them grow up very distrusting of authority, which is probably a good thing, but it's also making them distrustful in public health. It's just distrustful in, it's just the generation above them is just an oppressor. In their opinion, they see this, like, there's hypocrisy everywhere. They're forcing the kids to do all the stuff. They know the kids are already half immune because of the rate of this virus in the population, and they're still running after them with needles. What does it tell you about the generational exchange that's going on here?
Right.
It's an extraction process, and probably a wealth transfer problem as well, which is going on with all this inflation we're seeing as a result of this. So yeah, there's a lot of global issues that will have an impact on our business if we don't speak out. And so I guess there's some personal interest there.
Yep. Yeah. So I'm not sure what I say at the end of this video. I think there's a few more minutes left. I'm going to let it go. I apologize if I say anything silly, but clearly this was a wonderful use of my time to talk to him. I learned very much. I hope you did too. I think I just kind of say thank you and call him a hero at the end, but I'm not sure. There's a few minutes left. I don't know. I'm just really happy we finally got to talk. I'm so happy you waited. I can't say thank you enough.
Oh no, thank you for the work you guys did at Drastic. I mean, that was a really hot topic that I was afraid to touch too early on. That took a serious amount of you guys took a lot of risk on that, and I think a lot of people appreciate the work that you guys did there, because I can remember those days. Everyone was afraid that, well, what if we speak out about this and they take this and turn it into weapons of mass destruction and go attack at China? Like this, you don't want to be the guy that could trigger that, and you guys did not trigger that. I'm not saying you did. In fact, I think it's quite the opposite. If anything, it's been neutralized in that it's pretty clear the funding comes back to us. So there's equal blame to go around between nation states here. We shouldn't be encouraging the nation states to get jingoistic here. We got to clean up our own house here. We got too many labs that are leaking these things. And the virologists that are on this who are funded to continue this work are on a campaign trail to annihilate any lab leak stories, because they know that means they're going to get regulated like the nuclear industry. And frankly, the way they're behaving, they almost should be. I'd hate to say that, but
you should say that. I would say that definitely.
Yeah, they're out of control and arrogant, and we're seeing leaks left and right. I don't know if you saw the paper from the Venter Institute. Actually, it was from Beijing, accusing the Venter Institution of leaking SARS-CoV-1 in 2007, 2012. Right? I mean, if that can happen at their lab, and they've done excellent work throughout time, it can happen anywhere, and we need to up the bio-level safety levels, the oversight there when you're handling respiratory viruses that can have this type of global pandemic.
Yep.
In fact, I think it's just it's too loose right now, and you're not going to hear it from the mouthpieces on TV. They're going to pull in the people who benefit from this stuff, and they're going to tell you everything's fine. It came from pangolins, and they unfortunately, because they have the funding, they have the microphone right now. But hopefully the tide is turning with all the outreach that you guys are doing on this.
So that's Kevin McCarran. Kevin McCarran is a molecular biologist with about 30 years of experience, if he's honest, and he's worked on a wide variety of extremely impressive projects, has patents, and is far more experienced in the molecular biology space than I will ever be. And it was a privilege to talk to him. I am indebted for him to take that hour to talk to me over the weekend, and I hope we can have him on for a live stream very, very soon. So yeah.